So, hello everyone. I am Professor Andriana Margariti. I'm working in Queen University in Belfast in the Wellcome Wolfram Institute for Experimental Medicine. We are interested on diabetes and vascular complications. So, as you may be aware, diabetes is a devastating disease. There are very large numbers around the world of people suffering from diabetes. So it's really a global threat. The numbers are really increasing, and the years to come we are going to see very high numbers of people diagnosed with diabetes type 1 and mainly type 2. And what we're really interested in is to understand how diabetes is developed, but also to understand diabetic complications. And when I'm saying diabetic complications, I'm referring to vascular diseases. So we see people with diabetes develop complications, for example, problems in the eye, like diabetic retinopathy, also in the heart. In fact, 1 in 2 people with diabetes diagnosed with myocardial infarction and heart problems. So our research is really focused on understand these diabetic complications, and actually if we have better knowledge how diabetes is developed and how these complications are occurring, then we are going to be in a very strong position of developing new therapies for these people. So this will have an enormous impact of really improving the quality of these people.
So the principal focus of our work is research, and when we started 60 years ago, our intention was to make a big dent in the death toll arising out of heart conditions, cardiac arrests and heart events. And then about 7 in 10 people were dying of heart conditions, and we wanted to make a change. And over the years and over all of that research, we certainly have made a change, because now typically 7 in 10 people survive. There are about 100,000 people in Northern Ireland suffering from diabetes, and there is a real chance that as this science continues to be tested, that will make a difference.
And we have identified a gene that this is an RNA binding protein. The name of the gene is quakin 7, and we have found that this gene is highly expressed in blood vessels in diabetic patients. When this gene is highly expressed, then start damaging the blood vessels, and we have identified exactly how this process is working. By having this knowledge, we can go one step further and identify new treatments. So this is exactly what we are doing at the moment. So we have identified a few drugs that actually these drugs have the potential to low down the expression levels of these genes.
Undoubtedly what she has discovered, and while she's coming at it from a diabetes perspective, she knows that people, particularly with diabetes, can have heart and circulatory disease. So discovering that gene specifically affecting there and being able to potentially to switch that gene off is going to by default make a big difference to those with heart and circulatory disease. Even though they enter that journey through diabetes, it will undoubtedly make a difference.
I'm interested in science. Very early, young age, I was fascinated about the power of science. So I used to read medical and scientific books from the age of 10. And then by finishing the high school, I knew very well that my career will be on science. So then I proceeded with my undergraduate degree, and then I did my master degree in hematology, and actually there is where I first exposed to stem cells. So I was fascinated by seeing how this small cell has the potential to differentiate and make any cells within the human body. So then I did a PhD in cardiology and cardiac defects, and actually there I started using the stem cells and see the power of that. I came here to Queen University Belfast in 2013. I was before working in King's College London. So when I came here, my vision was to set up the cell reprogramming and actually to bring this technology here in Belfast. And actually I can tell you that we are the first and the only group at the moment working on this induced pluripotent stem cell technology that actually we can really develop these human models from diabetic patients here in Belfast. and particularly we are leading the field of endothelial differentiation. So we have spent many years really working hard, and the team here establishing the protocols for endothelial specification. So we have now these human models, so we can make these vascular cells, and actually a specific name for these cells is endothelial cells. This is the cells actually that they align in the blood vessels. So if you think the blood vessel is like a tube, so the internal layer of these cells called endothelial cells. And actually now having this, we can really understand what is happening, how this high glucose is damaging these endothelial cells in that patients. So we have developed these patient-specific cells, and you are seeing endothelial cells derived from that patients. So this is a cell culture that actually showing how we culture the cells in the lab, and actually how we perform different assays. So this is specific cells that they are living within our heart, and actually using this technology we can have now patient-specific cardiac cells. So you are seeing cells that actually they have the potential to beat, and they mimic exactly what's happened within the blood vessels. So by developing these human models, this is a great tool, a remarkable tool for drug screening. Because think about if we want to screen 500 or 1,000 drugs that actually they have the potential to treat diabetes and diabetic complications. You cannot really have a person with diabetes and screen all these drugs, because some of the drugs maybe are toxic or they are not working. So you need a good model in order to validate and actually get very reliable data that is going to be translated in the human settings. Exactly the same settings, the same cells, the same blood vessels that they will receive the treatment within the human body. So this approach really moved the field forward, because now we can be very close to the clinical settings by having this human model related to the disease. So we call them a phenotype of the disease in a petri dish. So we call them that actually you can make even an organ on a small setting, and we are really interested of making hearts, or brains, or make kidneys. And now the technology is here, so it's not something that is going to happen 20-30 years later. It's happening as we are talking. So we can use this approach by starting with a very small amount of blood sample, and actually we can create not only cells, we can create tissues, but also we can structure the tissues in a way and put them in appropriate scaffolds in order to create a heart and a kidney, a brain, or any other organ from the human body. Also, we are very interested to really support the next generation of scientists. Also, the students in the schools, but also in undergraduate degrees, we really want them to expose them on the research field, and also to have the opportunity to come and see what actually research is about, and to explain to them and to see in real life what they can do if they visualize themselves to become the next generation of scientists. Also, what I have seen the last few years, and it's really encouraging, is that many females are really feel supported, and actually they make a decision to pursue a career in science. So we see that these numbers have increased, and this is based on the supporting environment, and also females now feel more confident in order to really follow the path of science. Also, we have seen that many women feel strong and supported in order to apply for more leadership positions, as before we can see that this were mainly dominated by the female by males, but actually now we see that females, they take part, and they really actively contributed to these leading roles. That is very encouraging for the future, and it's something that we support, and we will make sure that in the years to come we will see even more female scientists join the field and actually make amazing contribution in the field. I always been fascinated of the power of science, because as I explain to my research team every day, science is very exciting because not every day is the same, but also you are doing things and you have the freedom to think and to really design experiments and think outside the box to create and find something something new. So there are people out there waiting for treatments. There are people that the quality of life is really impaired because currently there are no treatments to help that people really cure their diseases. So this is a very strong motivation for us to work endless and actually to be there to make sure that we understand the pathology of the disease, and actually we are creating that tools. We test the tools, we do the drug screening, we are creating functional cells to repair the damage that is happening to the bodies of that patients. That actually currently there are no treatments and they're just waiting. So strong motivation, and I really feel so passionate about that to work very hard and actually make a difference on that people's life.
This type of research, and when you see the people involved and you tour the labs, as we often do, not just with the researchers but with people who have been affected, and you see the joy almost in those that have survived, recognising the value of what people like Andriana's work can deliver. I'll tell you a little story. When I went for my interview for the British Heart Foundation, and we were concluding some details, and I was asking the one of the senior executives about the role, and she said, it's a job where when you're sitting around the table and you look up, you see everybody else crying too. And that's because there's been an achievement, or they've recognised the language of an individual, or there's been a contribution from somebody that really touches at your core. And that's the fuel for me for this job. And I know when I see it every day in people who've survived, families of those who've lost loved ones, but the determination seems to be there. Even in loss, people want to see a difference. With success, people want to see further success. And that, if you like, magic ingredient is really part of what the BHF is for me.